What's up, baby bio penguins? It's Friday, FRQ Friday. Yeah. So y'all realize we have like 5 days before the AP exam. So this is our last FRQ Friday. We're going to go over the 2 FRQs from the short FRQ. So 2021 number 5 and number 6. So we'll just do that real quick. And then after that, we'll log off. And also a reminder, if you don't know about it, I'm going to go live tomorrow on Marco Learning's YouTube channel to do a cram session. So they want me to do one the night before the AP exam, but I honestly believe that you should be sleeping the night before your AP exam, and you should be kind of focusing on relaxing and not stressing trying to do a cram session. So if you want to watch the cram session again on Tuesday, you're welcome to, but I will be going live tomorrow at 4 p.m. Eastern with Marco Learning to do a quick little cram session. I haven't really designed what I'm going to do, but that's what I've come up with. So let's get into our FRQ Friday before further ado, because you all love my FRQ Fridays, don't you? So today we're going to do 2021 number 5 and number 6, and ooh, now we have weird light coming from behind me. Okay, so I kind of made these a little small because I was trying to get it all in one slide. So the annual plant, went past it. Annual plant cycle, complete their life cycle, including germination, seed production, and death within 1 year. So basically it's grown and then it dies within that 1 year. There's a certain giant ragweed is an annual plant that rarely colonizes any land that has a disturbance such as plowing. The plant is considered an invasive species in regions outside native range. In particular, the seeds of the A. trifida germinate between early March to the end of summer, while the seeds of the other annual plants require warmer soil temperatures, thus they germinate in late April through the end of the summer. Researchers studied the influence of it on the biodiversity of other annual plant species that grow in the same field. In the spring, researchers marked off identical plots in a field that had been plowed in the previous fall and not replanted. All plants that grew at one half of the plot were left untouched, while all germinating were removed from the other half. So this kind of shows you what those two figures are going to look like in terms of like what they did for their experiment. Okay. So as we've seen before, part A is going to ask us some type of biology question. So describe a cause of logistic growth in ragweed populations. So we say, okay, well, logistic growth. Logistic growth is the one that has the carrying capacity, right? And so this has to do with that there must be some type of substance, whether it's a density-dependent factor like a disease or predation. There's something that keeps your population in check. It could also be limiting resources. So if you have a limiting space, limiting food, water, nutrients, since we're talking about plants, all of those can be things that limit it. So it says describe a cause of the logistic growth. It could be just that there is some type of limiting factor in our population. So the factor becomes limiting, which would cause population size to stabilize. You could also talk about any of our density dependent factors. So like space, sunlight, herbivory, phosphorus, nitrogen, and other density dependent factors. Real quick before we move fast, because of course we got to make connections to other things, right? Why is nitrogen so important in terms of growth of organisms? So if you remember, nitrogen is found in your amino acids as well as it's going to be found in your nucleic acids, right? So the amino acid has it in the amino group, while the nitrogen is going to be found in the nitrogenous bases of our nucleotides making up our nucleic acids. So it's important that we have nitrogen for that. In terms of phosphorus is needed for your nucleic acid. So you can't make any more DNA and you can't have the cell growing and dividing if it doesn't have more DNA, and we need phosphorus for that DNA. So of course phosphorus would be another limiting reactant that we would find. So our next question, or part B, again, is going to be looking at something with our experiment, right? So based on the representation in figure 1, explain why the scientists claim that plot B would be more resilient than plot A. So if we look at our diagram, and I need to move my camera a little bit. Okay. You can kind of see that there's like mostly the A trillified in this one. There's a couple of this little triangle, right? But we have mostly these X's. And then over here in plot B, I see, what is that? 4 squares, 4 circles, 4 triangles. So I see a large kind of diversity, right? You've got 3 different organisms. You have more species richness. And so though there's a more abundance of the trillified over here, there's a higher biodiversity in this group, right? And so if there was something that was to change in our environment, right, we have a higher chance of these organisms being able to survive because there's so much diversity. And so they asked, why is it more resilient? It's more resilient because it has a greater biodiversity. There's more species diversity in plot B than it is in plot A. And so I'll flip the screen so you can kind of see that part of the rubric. So plot B is more resilient because it has a greater diversity than plot A. And as you're practicing these FRQs, remember that anything that's in parentheses is a quote unquote optional statement. So you don't have to say those things, but it does help in explaining your information. But so all you had to say was that, one, it would have because it asked you in there, right, why plot B would be more resilient. You could say because it has a greater diversity in plot A. Okay. Ooh, one of my own students is here. That's exciting. All right. So on to our third part, right? So part C is going to always ask you to draw something, right? So we're in question 5, and question 5 always asks some type of drawing. So whether it's a pedigree, hey, the human geo guy. If y'all are taking human geography next year, don't forget to check out the human geo guy. I think you already had your AP exam this year, so it's not going to help this year, but next year, if you're taking human geo, make sure you check them out. Yes, you always have to write in complete sentences. Somebody just asked in the chat if we need to write in complete sentences. Yes, always write complete sentences. Even if it says identify, write it in a complete sentence. The concentration is like give it a full sentence to ensure you get that point. Back to the FRQ. So part C in our 5, is going to be a drawing, right? So it could be a pedigree. You could be drawing something going through a cell. You could be identifying parts of your cell. There's some type of manipulating of some type of figure, right? And so our part C today is going to give us information, right? So it said in the third group of plots, the researchers removed all of the seedlings of all the plants that germinated before June 1st. So basically we had an empty plot. And they want to know all the plants that germinated after June 1st were left untouched. So they say using this template provided, and then the different symbols we have. So you have an X, a circle, a triangle, and a square. Represent the expected plant species, and we should not draw more than 12 symbols. And so we have to assume all environmental conditions, right? Well, if you remember from the original experiment, it talked about that they would germinate like the trilla, or was it the the A trifeta would germinate early, right? So I talked about germinating early, and I said all the other annual plants needed to have kind of more and more conditions, right? But it said that both of them are going to continue to germinate into late summer. June 1st is not late summer. So what we're seeing is that these organisms are going to be able to germinate even after we've taken everything that germinated before June 1st. Everything else can still grow, okay? So you would actually expect through your diagram to show your circles, your squares, your triangles, and then you would also find X's. So they would both be there because both of these organisms, the annual plants as well as the invasive species, all these organisms are able to germinate into late summer. Okay, and so this was actually like what was on the scoring guideline. They said all 4 species must be added. You didn't have to have them in equal quantities. You just had to have all 4 of them on there. Okay, that way you would be able to demonstrate that you understood that all of the organisms would be able to grow. Okay, and then part D. So we're supposed to explain how invasive species such as ragweed would affect the ecosystem biodiversity. So think to yourself like, okay, what is an invasive species? And then you have to connect the invasive species definition that you know to biodiversity. So this is more of that application component, right? So we know invasive species were not native to the area. They have come in some way, whether it has been a human bringing that invasive species in, or whether it has just naturally gotten there. But it has no predators. There's nothing to restrict its growth. So it goes through this exponential growth curve, right? It's got unlimited resources. It has no natural predators, and it just basically ruins the whole biodiversity, right? Because now it competes with things that it didn't have to compete with before, right? And it outcompetes those native species. And so we see all that fun stuff. So here's what it actually says. It says an explanation showing a process of relationships. So you have to talk about how the biodiversity decreased. Different examples, you could say that there are no predators for the invasive species, so the population grows faster and faster, which reduces that biodiversity. You talk about the invasive species germinates earlier, uses up resources, and reduces biodiversity. So this would have been something you could have pulled from the prompt, right? So our prompt told us that it germinated earlier, so you could connect that to the specific question here and say, okay, well, the invasive species must germinate earlier, so it gets all those resources before the native species are able to, and so we won't have as many of those native annual plants, right? Or you could say that it outcompetes the other species, and that is going to reduce biodiversity. Overall, you have to talk about the connection, right, between the invasive species and what that does to biodiversity. Okay, so if you notice, every single one of those options all talk about that we reduce biodiversity. Okay, so you have to talk about the effect. This is a cause and effect connection. Okay, this isn't just let me name drop something. This is some type of application. Okay, and then part number 6, right? Number 6 is a little complicated on this one. And again, I made the font really small. So give me a second to get through all of that the question prompt for you. As you notice, it's just tons of reading on these exams. So we have a small invertebrate krill species that's adapted to cold seawater. Over the past 10 years, there's been a gradual increase in our water temperature of the krill habitat. Sustained increase in water temperature is ultimately going to affect the ability of the krill to survive. Of course, you know that if it gets hotter, then of course it goes through denaturation of its proteins, which is a problem, right? So one effect of higher temperatures is protein misfolding within cells. Krill have several HSF genes that encode for the heat shock proteins. These proteins help prevent protein misfolding and help to refold. So basically all these HSF proteins, their jobs are to refold these proteins that are getting denatured, right? And so in this high temperature, we would expect there to be more of those HSP proteins, right? So the scientists conducted experiments to detect changes in the expression of the genes when the krill were exposed to temperatures above 4 degrees. The experimental group of krill was maintained in tanks at 4 degrees, placed in tanks with 10 degrees for 3 hours, and then the krill were given a 6-hour recovery period in the 4-degree seawater tank. Okay, so basically we have it in 4 degrees, we heat it up, and then we put it back in 4 degrees, right? They then analyzed the expression of 3 different concentrations of mRNAs, 1, 2, and 3, which gets really complicated in a second. Transcribed the certain HSP genes in both the heat shock krills. Do y'all feel like I'm talking gibberish right now? Like, how many of you have heard of any of those words that literally were just mentioned? Okay, so that's going to happen on the exam. You're going to read a question and just go, what did I just read? Like, what was that? And sometimes you're going to have to reread, right? Sometimes you're going to have to go back through that prompt. So I recommend that as you're reading these questions that you underline, you circle, you draw some type of visual that helps you to see connections to things if need be, okay? And then of course we have that graph, then you can kind of see what observations you see there, okay? So first part, so we need to identify HSP mRNA that has the slowest rate of concentration increase in response to the heat shock protein, right? So we need to look at our graph, and it says identify the one that has the slowest rate of concentration increase. Well, rate is just slope. So we're looking at which one has the smallest slope. So as you see here, we've got the circles, the triangles, the black triangles are going to increase. So that does not show a slow slope, a small slope, right? And then we can see here the squares, they go up and then back down. But our circles kind of stay basically along the bottom. And our circles correspond to mRNA 3. So we kind of click there and say, okay, yeah, that is our answer. Is mRNA 3 would be the one that has the slowest rate of increase or the slowest slope, because it is the one that stays basically level across the bottom, right? And slope is just your rate, right? And so someone just asked the question, if you can write all over the FRQ booklet. Yes. There's going to be 2 booklets. You'll have an FRQ book that has all of your questions in it. And then you'll have a writing book, like an answer book. And that's where you'll write all of your answers in it, is on there. And I somehow lost the entire chat. There it is. So you'll write in the FRQ booklet that has all the lines in it. So you can write anywhere you want on either one of them. The only thing we will score is whatever's within that square. So there's like a square that you're going to have on your sheet in which you write your answers. If you write outside of that box, we cannot see it because they're being scanned in. Like, we won't see those stuff. It has to be within the box. Please make sure it stays in the box. Okay, so on to B. B says to describe the trend in the average concentration of mRNA-1 throughout the experiment. So we're looking here at the black triangles, right? So I want to see, well, what do they do? Okay, so from 1 to 3, it stays about level, right? And then from 4 to 6, it looks like it has a slow increase. And then from like, I don't know, what's that, 7 to 10, it has a very steep increase. And then from like 10 to 12, it decreases. And that's literally what they wanted you to write. There seems to be no change in concentration from 1 to 3, increased concentration slightly between 3 to 6, and then we have an increased concentration at a greater rate from 6 to 10, and then a decreased concentration after 10. So you're literally telling them what is happening to your graph. That is what they actually do. They said, describe the trend. Tell me what happened in my graph. And so this involved you being able to find the correct line. So can you read a legend to figure out which one was mRNA-1? And can you appropriately tell me what you see in the graph? So not too crazy there. So part C, scientists hypothesized the heat shock proteins translated from mRNA provides a greater role in refolding proteins than does HSP translated from number 2. And it says use the data. So again, we have to look at our graph, right? So we had the heat shock take place here. After that heat shock, right, in this little 4-degree area right here, what do we see is the difference between mRNA-1 and mRNA-2? Well, mRNA-1 increases, while mRNA-2 decreases. So what that tells me is that even though the mRNA-1 is in a higher level, it's showing me that this means that they need to be refolded, right? Oh no, sorry, I was trying to flip the camera. So it's showing us that the mRNA is more essential in making that refolding because it's still being expressed and still being used after we've had that heat shock. So we're trying to reform those proteins versus mRNA-2, we see that decrease because it's no longer really going to refold those proteins for us. Okay. In our very last section, part D, mRNA-1 and 2 are transcribed from the same gene. Explain how a cell can produce 2 different mRNAs from one gene. So how can I have the same DNA but get 2 different pieces of mRNA from it? So you have to think, this requires some biology knowledge, right? How can I get multiple strands from the same DNA? Well, there's introns and exons, right? So you can go through alternative gene splicing, cut out some of those exons, of course, all the introns, which will give you different sequences, right? And so that is one of our ways. I think there may be other things that they have on there. So the other options you could have put are you could have different transcription, termination sites. So different poly-A sites. You could have also said different promoters. And so if they have a different promoter, remember RNA polymerase will bind to the promoter to allow for the making of that mRNA. So there's a way that we could actually, I guess, have different promoters, which led it to be different. So there we go. Anyway, so I hope that was helpful to everybody. Quick reminder, I am going live tomorrow on Marco Learning's YouTube channel. So that'll be tomorrow, 5/7 at 4 p.m. Eastern time. I will be at my school because I have no internet at home. And so I will see tomorrow. We're going to do a cram session. My current plan, I don't know if I'm actually going to do it or not because I haven't made the PowerPoint yet because, well, that seems to be my MO is I'm running around always never having time to do anything. So I'm planning on doing a general, like, here's what the exam structure is. I'm then going to try to do a quick review of each unit and then do like a question or 2 for each unit. So not the questions that I've already done in the other Marco sessions, try to find a new multiple choice question or a new free response question to kind of quickly draw home the information so that y'all can either see, error bars. We could talk about those. We could talk about, chi-squares. We could talk about the different, biology that's needed. So if you're needing a little quick cram session, tomorrow, 4 p.m., it will be recorded. So if you can't catch it at 4 o'clock, not a problem because it's recorded and you can watch it anytime, as well as the other 7 videos that I did with Marco Learning. It's all posted on there for you. So good night. And remember, AP Bio Penguins address success. Bye, y'all.